So today we're diving deep into the world of modern drug design. We're talking about a place where scientists are fighting diseases like cancer on a scale that's, well, it's literally invisible. They're basically building microscopic weapons one molecule at a time. Let's get into how they pull it off.
And really, this is the fundamental question, right? Imagine you have to make a key that fits perfectly into a lock, but that lock is thousands of times smaller than a single grain of sand. That is the incredible puzzle that scientists are trying to solve every single day.
All right, so our focus today is all about taking on one of the biggest challenges in medicine. You could say the battlefield is the human body itself, and our weapons, they're not much bigger than individual atoms.
And the specific problem we're tackling here is lung cancer. For a long time, treatments were kind of like carpet bombing. They worked, but they caused a ton of collateral damage. The goal now is totally different. It's to design these molecular smart bombs that are super specific, targeting only the cancer cells while leaving the healthy ones alone. Every good mission needs its heroes, right? Well, in this story, they're not people, they're molecules. We've got 3 potential drug candidates, all created in a lab with the hope of becoming the next big anti-cancer agent. So here they are. Let's call them compounds 2A, 2B, and 2C. These molecules were built using a specific chemical framework, an acridine dione scaffold, which has already shown some real promise in fighting cancer in the past. But what's really interesting is just how similar these molecules are. Look closely. Molecule 2A, that's our baseline. Then 2B has this tiny little nitro group tacked on, and 2C has a chlorine atom. This isn't random. It's a very deliberate, very controlled experiment designed to answer one crucial question. How can one tiny atomic change completely alter the behavior of a potential drug? Think about it. Before you can know if a key fits a lock, you have to know the exact shape of the key. That seems obvious, right? So the very first step for these scientists was to create a super precise 3D blueprint for each of their molecular candidates. And they do this with an amazing technique called X-ray crystallography. You can basically think of it as a CT scan, but for molecules. They shoot X-rays at a crystal made of the compound, and by analyzing the pattern of how those X-rays bounce off, they can map out the position of every single atom. It gives them a perfect 3D model. And here's what they found. The analysis confirmed the exact 3D shape for all 3 molecules. You can see they're not flat. They have this specific bent structure that chemists call a half-chair conformation. But the most important thing here is we now have the exact blueprint for our keys. Step 1 is complete. But, a molecule almost never acts alone. Inside the body, it's swimming in a sea of billions of other molecules. So just knowing its shape is only half the battle. We also have to understand how it interacts with its neighbors. It's kind of like mapping out a molecule's social network. To do that, scientists use another incredible visualization tool called a Hirschfeld surface. This thing is basically a heat map that gets wrapped around the entire molecule. And those bright red spots, those are the hot spots, the areas where the molecule is interacting most intensely with its neighbors, almost like it's reaching out and shaking hands. So let's take a look at the Hirschfeld surface for our baseline molecule 2A. You can see a few clear red hotspots, right? These are showing us where the molecule is forming weak bonds with its neighbors. This is its normal, baseline, social behavior. Okay, now watch what happens when we add that tiny nitro group to make molecule 2B. Whoa, the map completely changes. All of a sudden, we have this whole new set of super intense red hotspots, and they're all related to the oxygen atoms in that new group. That one small tweak completely rewired the molecule's entire social network. And the numbers back this up big time. For the original molecule, about 17% of its surface interactions involved oxygen, but for molecule 2B, that number almost doubles to over 31%. That is a massive shift in how this molecule behaves, all from just adding 3 little atoms. So, why do we care so much about how molecules shake hands? Well, because these interactions, they determine pretty much everything. They control how a drug packs itself into a pill, how well it dissolves in your stomach, and how easily it can get absorbed into your bloodstream to actually reach the cancer cells. Understanding this stuff is absolutely key to designing a drug that works. Okay, so we've got the 3D shape of our keys. We know how they like to interact with their surroundings. Now it's time for the ultimate test. Do they actually fit the lock? It's time to run a virtual clinical trial right inside a computer. Now, The locks in this case are very specific proteins known to make lung cancer grow and spread. Two of the most infamous ones are called EGFR and ALK. In a lot of cancers, these proteins are basically stuck in the on position, just telling cells to divide over and over. So the goal for our molecular key is to fit perfectly into these protein locks and just jam them, shutting them off for good.
This virtual test is called molecular docking. Here's how it works. Scientists take the known 3D structure of the cancer protein, that's the lock. Then they take the 3D structure of our candidate drug, the key that we just figured out. Then a supercomputer gets to work, simulating millions of ways to fit that key into the lock, and it calculates a score for how snugly it binds. And the results were fantastic, incredibly promising. The docking simulations predicted that all three of our molecules, 2A, 2B, and 2C, could bind really effectively to both of those big cancer targets, EGFR and ALK. The virtual trial was a success. The keys fit the locks.
So let's zoom out for a second. What does all of this research really tell us about the future of medicine and how we're going to create new drugs from now on? What we've just walked through is basically the modern drug discovery pipeline. It's this beautiful, rational, step-by-step process. You build a molecule, you map its 3D structure, you visualize how it socializes, and then you test it virtually. All of this happens long before you even think about moving into expensive and time-consuming lab trials. And here's the real game changer. We don't have to rely on just luck or trial and error anymore. By using these powerful computers and visualization tools, scientists can literally be molecular architects. They can see into the invisible world of proteins and chemicals and design smarter, more targeted, and more effective drugs, starting from the atom up.
And that really brings us to a final thought. We've seen how this approach can be used to target proteins that drive lung cancer, but these same principles can be applied to countless other diseases that are caused by faulty proteins, everything from Alzheimer's to antibiotic-resistant bacteria. So the question isn't really if we can design these keys anymore. It's what lock should we choose to open next.